Hello, good afternoon, everybody. I'm going to be telling you about two families of compounds that we have discovered. They're aminosterols, and we're calling them hormones. And they're really quite relevant to a longevity meeting. We believe them to be present when we're young and to disappear as we get older, as do many hormones. The first family of compounds Michael discovered in the shark a number of years ago, and we're using synthetic derivatives of these, of course, for the human studies. And of that group, they're made in the shark liver. Of that group, we are developing Ento-1, which has a profound effect on misfolded proteins. It prevents alpha-synuclein, for example, from aggregating and dislodges existing aggregates from enteric nerves, restoring neural signaling from the gut to the brain. And we have data from phase 2b studies in 200 patients with Parkinson's disease. And we've shown to date improvement in constipation, dementia, psychosis, depression, sleep, REM behavior disorder, and circadian rhythm, with continued improvement beyond the treatment period. And on the right is the first of the compounds from the second family that we have recently isolated from mammalian brain. And we're developing Ento-3, which has a profound influence on insulin resistance. It acts centrally at the level of the brain, and we're going to be developing it for the treatment of obesity, diabetes, and Alzheimer's disease, and clinical studies with that are due to begin in just a couple of months. So to spend a few minutes on Ento-1 in Parkinson's disease, Ento-1 reverses the neurological symptoms of Parkinson's disease. Parkinson's disease, as we have shown, begins in childhood, in the gut, with GI infections, with the production of a defensive protein that we call alpha-synuclein. This protein is produced in the nerves of the gut. It helps confine the infection. It calls in the immune system, and it also alarms the rest of the nervous system as to the existence and location of an infection. And in most instances, this works brilliantly. The infection is confined, and then alpha-synuclein levels progressively taper over the next few months and disappear. Except in some individuals where they don't, either because of resistant infection or recurrent infection or failure to confine it, or a funky white cell immune system, or some underlying genetic abnormalities that fail to clear the alpha-synuclein. And in those few cases, as occurs, for example, in children with autism, and in eventual patients with Parkinson's disease, the alpha-synuclein accumulates, clogs up the gut cells, they fail to communicate with each other, constipation supervenes. And as it spreads, communication between the enteric nerves and the brain via the vagus nerves is muffled, and cerebral symptoms develop, starting with circadian rhythm abnormalities, sleep abnormalities, and mood and apathy, and eventually leading to motor symptoms and then psychosis and dementia beyond that. So how does Ento-1 work? It's taken orally. It acts locally at the level of the gut. It penetrates the epithelium. It gets into the enteric nerve cells, and it's very positively charged. So purely electrostatically, it displaces all kinds of misfolded proteins, including alpha-synuclein, from nerve cell membranes. Here is a cell with green fluorescent alpha-synuclein against the membrane. And you'll see when we put a couple of drops of Ento-1 onto this, within seconds, boom, all the alpha-synuclein aggregates are displaced off the membrane and into the cytoplasm, allowing for faster clearance. About the same speed, the function of the enteric neuron is restored from not producing any action potentials when it's zapped to producing multiple action potentials, firing like a normal enteric neuron when we place a few drops of Ento-1 onto this. And as a result of normal function now of the enteric nerve, they can now communicate with each other, and that's how constipation is overcome. They can also amplify the specific signals going up the vagus nerve to the brainstem and the hypothalamus, and have an effect on the brain. And the effect they have on the brain is very dramatic. This is within a CFOS activation within 2 hours of oral ingestion of this compound. And in red, you can see the areas of the brain that are stimulated. And these have to do with what we call the paraventricular thalamic nuclei that are engaged in awakening, alerting, attention, focus, and circadian rhythm, and also executive function and cognition. And in green, all the areas that are inhibited which include vast areas of the occipital cortex, the visual cortex, the olfactory cortex, and a whole host of subcortical structures that have to do with visual hallucinations. So if you ask me, what do you think, how do you think a symptom with Parkinson's disease might improve with this? I would say, well, the patient will be more awake during the day, better sleep at night, won't have REM behavior disorder, will have better attention, better focus, will concentrate, will have better memory, cognitive function will improve, dementia will clear, and hallucinations will disappear. And this is, in fact, precisely what we are seeing in the clinical studies. So we first conducted a 50-patient phase 2A study that we call RASMET, long since been published, in which we took patients with Parkinson's disease and constipation and treated them for 3 weeks with an ascending daily dose and then fixed them at a prokinetic dose. We escalated until they got proper bowel function on a regular basis, and then we kept them there for a few days. And all these symptoms that I mentioned earlier improved, specifically dementia and hallucinations that were almost completely eliminated. So we went on to conduct this phase 2B study, what we call INCARMED. It's to be published in the Annals of Internal Medicine in 3 weeks' time. It was a randomized version of the first study. So we took 150 patients now with Parkinson's disease and constipation, and we randomized them. We also stratified them to start at low or high dose, depending on how constipated they were. The primary endpoint was recovery of bowel function, the lowest hanging fruit. We escalated the dose to a prokinetic effect, and then we held them there for the remainder of the 3-week period. And the primary endpoint was statistically significantly better in the treated group compared to the placebo group. And this benefit persisted for up to 6 weeks following treatment discontinuation. All our secondary endpoints were met, highly significant at the end of the treatment period, and that improvement continued beyond the treatment period for about 6 weeks. Hallucinations improved and disappeared in the treatment group compared to the placebo group, where all the high scores at beginning remained. And dementia improved in the group as a whole, and benefit persisted for 6 weeks beyond the treatment period. And specifically in the group that were demented at baseline, where they improved even further. So in summary, we showed that there were no safety or tolerability issues. The side effects were confined to the GI tract, as you might expect from a drug that's not absorbed into the systemic circulation. The primary and secondary endpoints were met, as were the exploratory efficacy endpoints in cognition, hallucinations, and mood, and there was no worsening of motor function. So we essentially replicated the results of the first open-label study, and we believe that by addressing and rectifying the underlying pathology, Enterin produced an improvement in multiple non-motor symptoms with a persistence of effect beyond the treatment period, suggesting perhaps a modification of the disease process. And these are our upcoming studies. We intend to do a psychosis study, Parkinson's disease-related psychosis study, and an autism study, because it has actually features remarkably in common with Parkinson's disease. That's subject for another discussion. And of course, beyond that, the phase 3 study. A few words about ENTO3. This compound, as I told you, is present in neonatal mammalian brain and decreases with age. It increases insulin sensitivity at the level of the hypothalamus. It rapidly normalizes glucose and causes weight loss in obese diabetic mice, also with a persistence of effect beyond the treatment period. I don't need to tell this audience about how important insulin resistance is and how many conditions it affects, including Alzheimer's and Parkinson's disease. And of course, the best-in-class agents are GLP-1 agonists, which on paper sound great, but in practice don't always produce the glycemic control required, don't always produce the weight loss required. And a lot of people discontinue it because of the GI side effects, such as nausea predominantly. And so the unmet need is for something that is for a drug that can improve insulin sensitivity, that can improve glycemia and weight, at least as well as the GLP-1 agonists, but which doesn't cause GI side effects. And we believe ENTO3 to be perhaps such a compound. It has a very different mechanism of action than GLP-1 agonists. It inhibits PTP1B at the level of the hypothalamus and increases insulin sensitivity. A single dose given to obese diabetic mice normalizes blood glucose, brings it into the normal range. 5 doses given every 3 days over a 2-week period reduce body weight by about 20%, normalize glucose, halve insulin, normalize ghrelin, and normalize liver function tests, and reduce liver fat by a good 30–40%. And the effect on the brain is dramatically different than semaglutide, one of the leading GLP-1 agonists. In the case of ENTO1, about 40 areas in the hypothalamus and brainstem are activated within 2 hours of taking the medication, whereas in semaglutide, it affects about 12 nuclei, very few in the hypothalamus, and most of the areas in the hypothalamus that ENTO3 stimulates are the areas that are heavily involved in energy and appetite. And so to summarize, the differences between ENTO3 and GLP-1 agonists, ENTO3 targets PTP1B in the hypothalamus. Its primary effect is on increased insulin sensitivity, whereas GLP-1 agonists target the GLP-1 receptor, and a primary effect is on increased glucose-dependent pancreatic insulin secretion. In the case of ENTO3, weight loss is greater than would be achieved by reduction of food intake alone, and glycemic control is independent of weight loss. Blood glucose is rapidly normalized as a result of increased insulin sensitivity prior to any weight loss, which is not the case for GLP-1 agonists, where weight loss is proportional to reduced food intake and glycemic control is dependent on weight loss. And these are our upcoming phase 1 studies starting a couple of months from now. And just to finish off, I added these 2 slides in view of previous talks. ENTO3 has a profound effect on animal models of Alzheimer's disease. In very aggressive models, it eliminates inflammation, eliminates tau in the hippocampus and hypothalamus. It prevents neuronal loss. It reverses memory deficit. It normalizes behavior, dramatically increases healthy lifespan. And this is a survival benefit. The treated animals are all alive at the end of 15, now 18 months, whereas most of the untreated ones are dead. And treatment begins here well after the onset of the Alzheimer's disease. And I thought I'd finish off by showing you a video of an untreated animal, very elderly, placebo group rather. You can see its hind legs are spastic, they're not moving. It's got a big kyphus. It's put down in the cage, and it sort of just sits there. It's shaking. It doesn't really do very much. It's not exploring. It's not moving. You can see a marked kyphus. And on the right, a treated animal. Look at the back legs moving like crazy, wanting to be put down. No kyphus at all. It's been put down, and you'll see now just how much it's exploring the cage, how fast it's moving compared to the other one that's just staying put. And that's where treatment was begun 9 months into the animal's life, at which point they look like the one on the left. And they're now going strong at 18 months and looking none the worse for it. So with that, thank you very much for the opportunity to present.
Thank you, Denise. And it's always incredible to see the video of treatment as opposed to just a slide with static data. So kudos to you for including your mice participants. What is the oldest age of mouse that you've given ENT03 to?
We've started between 8.5 and 9.5 months in this PS19 model, where the histological changes begin at 3 months and the behavioral changes at 4 or 5 months, and the learning deficit soon after that. By 8.5 months, they have profound learning deficit, memory deficit, and profound inflammatory changes. And so we've treated from 8.5 months up to 18 months, and they're going strong.
Okay, so this is truly a reversal model.
Correct.
Wonderful. And congratulations on your recent round of funding, coming at a very, very good time.
Yeah, we could actually do some more. We need some more funding for the PD psychosis study. We've got enough money for the ENTO3 program to go forward, but we're now looking for money for the ENTO1 program for the PD psychosis study. So
how much are you looking to raise?
Well, as much as we can, minimum 15 million.
Okay. Okay. Well, wonderful. Well, we'll be watching and wishing you the best of luck. It's very exciting data.
Thank you very much indeed.